the mice were living longer and they were living more healthy. And so now we start thinking, okay, what are we going to do with the humans, right?
Naked mole rat's DNA could hold the key to long life.
Yeah. Naked mole rats live 40 years compared to 3 years for a mouse. Anything that is that size lives around 3-4 years. They've evolved some potent biological mechanism that is delaying aging.
Right.
And for the longest time, people actually thought they maybe knew what was happening, which is the cGAS protein, the cGAS pathway. We are eukaryotes, and so there's not going to be any DNA in the cytoplasm of the cell. Everything is going to be compartmentalized in the nucleus, which means if you ever see a DNA molecule in the cytoplasm, something has gone wrong. Either a virus has come in and infected you, or the cell is like straying into cancer territory. So the cGAS protein, the primary function is to detect that cytoplasmic DNA, bind to it, and then catalyze some reaction that activates an inflammatory response or an antiviral response. Now, that's great for the innate immunity part, right?
Okay.
It's detecting stuff in the cytoplasm, but it also has a paradoxical role where it starts detecting stuff in the nucleus of our cells.
Okay.
So this paper, it's unraveling the naked mole rat's genetic secret. They found the cGAS mediated mechanism in that naked mole rat, and they showed why that DNA, the part of the DNA that codes for the cGAS, is different than the humans, and how that difference actually manifests within the cell. What human cGAS is doing is it binds to the DNA, and then it kind of prevents all of the other repair mechanisms from coming in. What the naked mole rat version is doing is it binds to the DNA, and then it actively recruits all of the other chromatin binding aspects to like make this thing go faster, right? And have higher fidelity.
Instead of being an absolute firewall and blockade,
Yeah.
it sort of basically it's a little porous for specific helpers that it knows are actually there to facilitate a job function.
Yeah. The real nail in the coffin for me was like what they did was they could now introduce the mole rat version of this gene into model organisms like the Drosophila, and the fruit flies lived longer, and they were more robust, and they could climb up walls faster, right? In mice, the mice were living longer. Okay, what are we going to do with the humans, right? I mean, we can't genetically engineer, but you could always also have small molecule drugs that mimic what the naked mole rat version of the protein does. So let's say the human version has a particular shape, and we can take a small molecular drug that binds to that human version to make it mimic the shape of the mole rat. And then now it's going to do all the recruiting that the mole rat version was doing.
So it's sort of like we create a therapy that like mimics the cGAS process.